Today I will talk about soft source optical coherence tomography. It is almost similar to spectral domain optical coherence tomography setup, but there is a fundamental difference. In soft source OCT, there is a laser source which will vary over different wavelength over time. It will shift between low wavelength to high wavelength. Let's see how does it varies. Say this is your laser source. You can see that it has the reference arm or sample arm. It has the photo receiver. Now in spectral domain OCD setup, it's soft source OCD setup, wavelength sweep over time, and wavelength resolve interference recorded as a function of time. And here is the wavelength, and if you here a time resolve wavelength, and if you do the FFT, you can see the depth profile, and from there you can generate the image. So there are some advantages of soft source OCT over spectral domain OCT. It is less sensitive to fringe washout because the speed of the wavelength is pretty high, so you are acquiring data for a short period of time than the spectral domain OCT. So there is a less effect of fringe washout, and also a lower signal decay with depth. In our setup, we will use the AXN 1060 nanometer wavelength laser, and which will run at the sweeping speed of 100 kilohertz. So this is basically our setup. This is the laser source. We are sending the light here, and there is a beam splitter of 25-75, and 25% light will go to the sample arm here. Here you will put your sample, and then 75% light will go to this path. There is another splitter 1090. It will split the light. We'll send the 10% light through this path, and the 90% light to the reference arm path. This whole thing is called the Mach gender interferometer. It's used to take some data of calibration, because I'll explain a little bit later on why we did this. And in this part we can see some balance detector and low pass filter, and in the reference arm there is a neutral density filter here, and light from the sample arm is coming through this path, and also from the reference arm data is coming here. They are recombining here. There is an interference here, and it is passing through the balance detector and the low pass filter, and the data acquisition board is collecting the data, and it's going to the computer, and where you can process the data, or you can collect the data. So one important component of this is the balance detector. What it is doing is trying to detect the instability of the laser power. If there is instability of the laser power, it can remove the noise of laser power. It can reject. There is a one term called common mode rejection ratio. It can do that kind of rejection, and low pass filter is basically removing any high frequency noise. Same thing is happening in the sample arm. We are using the balance detector as well as low pass filter to serve the same purpose. In our setup, we tried two type of balance detector, one from the Thor lab balance detector, and from Newport balance detector. One thing for the MGI, it's important that it should be very stable. So to check the stability, we did that. So Newport one is Nirvana, and Thor lab BC we compare and check at which distance we find more stable, and we find that the Nirvana is more stable at about 1.5 millimeter path difference. Now, one thing to mention that In swap source OCT, we cannot regenerate the wavelength sweep same way every time. There is a small instability. This instability is due to the laser source. It can be come from the laser source, or it can be come from the interferometer or the data acquisition hardware. So, but if you wanna generate the data properly, you have to make sure that there is no instability in your data. So to remove all the instability, we need to use the calibration data that we can collect from here. We can collect the calibration data from the MZI, and using this collected data, we can resample the exact same wave number space simultaneously by using this calibration data. So we have to resample our sample arm data to match exactly same way we collected the data in the MZI, so that there is no phase instability. This is very important. Otherwise, we will not be able to do the phase resolve measurement as well as some other thing like fixed pattern noise removal. Your image will be noisy. So that's one of the important thing. Say here is some of the example that what will happen. So if you don't remove the fixed pattern noise, then so this is the image. You can see the vertical strip. But if you do the phase stabilization by the MZI data, then you can see that this is with phase stabilization. You can see quite a good data, and you can see the same thing, that what kind of artifact you have from the. This is the eye data. You can also see that the flow image or the intensity image, they are not they doesn't look good if you don't stabilize the phase. So it's important to collect the MGI data and do the phase stabilization by resampling the sample arm data to match exactly like what are the wavelength frequency, what are the wavelength you are getting every time from the laser source. You have to match that to get the better result. In the next video, I'll talk about the how you can do this kind of phase stabilization and do the dispersion compensation, and finally how we can see the primary image from the source OCT setup.